Hello, my name is Joshua Mugabi. I welcome you to Reproduction in Living Organisms. Questions 16 to 20. Distinguish between organs of vegetative propagation and organs of pollination plants. State the advantages and disadvantages of natural vegetative propagation. Outline the ways through which the variation can rise in a population. Distinguish between the organs of vegetative propagation and organs of pollination in plants. Organs of vegetative propagation are the parts of the flowering plants other than the flowers, such as the roots, stems, or leaves, those specialized in developing into new individuals when detached. For example, the rhizomes, where we have the ginger, the couch grass, canna lily, and spear grass. We have the culms, like in the cocoyam. We have the stem tubers, like the Irish potato, the yams. We have the root tubers, for example, the sweet potatoes. We have the bulbs, like the onions and garlic. Then we have the swollen tap roots, like the carrots. We have the stolons, like the blackberry, runners, like the strawberries. Organs of pollination are plant parts which are specialized in storing the food used to develop into new individuals, enabling the plant bearing them to survive adverse environmental conditions like drought. For example, the rhizomes, the culms, the stems, and root tubers, bulbs, swollen tap roots. Part B. State the advantages of natural vegetative propagation. 1. It is a rapid means of reproduction. The offsprings are genetically identical, preserving good strains. Pollinating organs enable survival in adverse conditions. Their dispersal and spread is independent of the external agents, hence the process is faster. Plants are less affected by environmental factors. Females pass all their genes to their offsprings. State the disadvantages of the natural vegetative propagation. It can lead to overcrowding and competition for nutrients unless separated artificially. New varieties cannot be produced except by mutation, resulting into reduced vigor and strength. Disease typical to a species are rapidly transmitted and can disseminate a crop. But C. Outline the ways through which variation can arise in a population. It can arise during fertilization, when the male and the female gametes with different genotypes they fuse to form a zygote. It can happen by mutation, during which there is alternation in gene structure and the sequence. During meiosis, by crossing over, during prophase 1, and also during random segregation of chromosomes on the metaphase plate. It can be caused by the effect of environment, may also prompt variation. Explain the processes during fertilization and state the significance of each. Sperm capacitation, a chromosome reaction, first block, cortical reaction. Part B, describe the hormonal control of spermatogenesis in humans. Explain the processes during fertilization and state the significance of each. We shall start with sperm capacitation. Sperm capacitation, it involves the activation of the mammalian sperm to fertilize the egg, during which the acidity and enzymes in the female genital tract causes perforation of the sperm head by removal of the cholesterol and the glycoprotein to allow the entry of calcium ions and the release of the acrosome enzymes. The entry of the calcium ion increases the beating activity of the sperm tail and also promotes the acrosome reaction to enable the sperm to penetrate the egg. Let's look at the process of acrosome reaction and its significance. Acrosome reaction occurs in the sperm head on making contact with the secondary oocytes, during which the cell and the acrosome membranes rupture to release hydrolytic enzymes. For example, hyaluronidase and protease. This enables the sperm head to penetrate the egg membranes. First block. First block, a process during which contact of the first sperm with the egg membrane is instantly followed by an electrical potential charge in the egg membrane to prevent entrance of any more than one sperm. The first block prevents entry of more than one sperm into the egg, which would be polyspermy. That would upset the diploid state of the embryo. Let's look at the cortical reaction. Cortical reaction occurs following the sperm penetration of the secondary oocyte, during which lysosomes, which are the cortical granules, they fuse with the plasma membrane and release their contents, causing the vitelline membrane to harden and form the fertilization membrane to prevent polyspermy. Formation of the fertilization membrane, this prevents multiple sperm entry into the egg, or what we refer to as polyspermy. That would upset the diploid state to cause the death of the mammalian embryo. Part B. Briefly describe the hormonal control of spermatogenesis in human. Interaction of hormones from the hypothalamus and the anterior pituitary gland working together control spermatogenesis. From the hypothalamus, a gonadotrophin-releasing hormone stimulates the anterior pituitary gland to secrete two gonadotrophic hormones. So we have the hypothalamus. The hypothalamus will release the gonadotrophin-releasing hormone, which will interact with the anterior pituitary lobe and it will cause the secretion of the luteinizing hormone and the follicle stimulating hormone. The follicle stimulating hormone and the luteinizing hormone, or the interstitial cell stimulating hormone, are the ones that are produced by the pituitary, anterior pituitary gland. The follicle stimulating hormone stimulates spermatogenesis, causing the Sertoli cell to complete the development of the spermatozoa from the spermatids. Follicle stimulating hormone causes the Sertoli cells to release a peptide hormone inhibin that specifically inhibits follicle stimulating secretion. When the pituitary gland secretes the follicle stimulating hormone, the follicle stimulating hormone is going to cause the Sertoli cells to complete the development of the spermatozoa. And then it will also cause the Sertoli cells to release a peptide hormone inhibin, which will inhibit further production of follicle stimulating hormone. The luteinizing hormone stimulates the Leydig cells, or what we call the interstitial cells of the testes, to secrete testosterone. Testosterone stimulates the growth and development of the germinoepithelial cells, those are the spermatogonia, to form the sperms, and also works with the follicle-stimulating hormone to stimulate the Sertoli cells. However, increased testosterone levels inhibit the secretion of gonadotrophin-releasing hormone and luteinizing hormone. So the luteinizing hormone, which is secreted by the pituitary anterior gland, is going to cause the Leydig cells. It will stimulate the Leydig cells. These Leydig cells will now secrete the hormone testosterone. Now the testosterone is going to stimulate the germinoepithelial cells in the testes to form the sperms. And then also it will stimulate the Sertolis with also the follicle stimulating hormone for the development of the spermatogonia to form the sperms. However, an increase in the testosterone is going to inhibit further production of luteinizing hormone, and it will also inhibit the hypothalamus from secreting the gonadotrophin releasing hormone. (18) Describe how the structure of the human gametes is related to function. And B, compare spermatogenesis and oogenesis in humans. Let's start with the structure of human gametes and their relationship to their function. We shall start with the ovum. Here we have our ovum. It has the nucleus, it has the cytoplasm, the layer zona pellucida and jelly coat. Inside the cytoplasm there are cortical granules, and outside the zona pellucida layer there is corona radiata, or what we call follicular cells. We have the yolk cytoplasm. This contains fats and proteins, and these ones nourish the developing embryo. That is the cytoplasm. Then we have the cortical granules, or the lysosomes. These ones contain enzymes that alter the structure of the vitelline membrane to prevent polyspermy at fertilization and avoid upsetting the diploid state of the zygote. So inside we have the cortical granules. These cortical granules, they contain enzymes that will change the vitelline membrane to prevent polyspermy. The vitelline membrane undergoes structure changes to prevent polyspermy at fertilization. We have the nucleus in the middle. This contains 23 chromosomes that complete meiosis II at fertilization to provide the female haploid nucleus. Then there are polar bodies. Polar bodies, they also contain the 23 chromosomes, but is non-functional and degenerate. Let's now look at the sperm. This is the structure of the sperm. It has got the head. It has the middle piece filled with mitochondria, and then it has the tail, which is the flagella. The sperm has got the acrosome, this front part here. The acrosome contains hydrolytic enzymes, and this facilitates the penetration of the egg membrane prior fertilization. So the acrosome here has hydrolytic enzymes which will help the penetration of the egg membrane during fertilization. Then there is the nucleus, which is in the middle here. The nucleus contains haploid set of chromosomes. Those are 23 chromosomes, which on fusion with the egg, they restore the diploid state of an organism. It has got the mitochondria. Mitochondria, they are in the middle piece here. And these complete aerobic to release ATP required for the contraction of the filaments during the sperm movement. So the mitochondria that we have in the middle piece carries out aerobic respiration and release energy, which is used by the filaments in the tail to contract and bring about the movement in the sperm. There is the tail piece, or what we call the flagellum. This tail piece, all of this one here is the tail piece. Sometimes it's called the flagellum. It enables the mobility of the sperm, the movement of the sperm. One pair produces microtubules that form the axial filament of the flagellum. Part B. Compare spermatogenesis and oogenesis. We shall start with the similarities. Both begin with the diploid germinoepithelial cell. So in the formation of spermatogenesis, the process of oogenesis, they both begin with the diploid germinoepithelial cells. There is mitosis and meiosis, which are involved in both of them. Both of them, they yield or produce haploid gametes, which are half the number of chromosomes of the parent, and they both occur in the gonads. Spermatogenesis occurs in the sperms, testes, and oogenesis occurs in the ovary. We look now at the differences. In spermatogenesis, this occurs in the seminiferous tubules, whereas oogenesis occurs in the ovaries of the female. Spermatogenesis begins only at puberty, whereas oogenesis begins during embryonic development. So genesis takes place when the embryo is being formed, but spermatogenesis will take place when the individual reaches that stage of puberty. Spermatogenesis is a continuous process and occurs all the time, meaning that when it starts, it does not stop, it is always continuous. Whereas oogenesis is a discontinuous process. Only one egg matures in about 28 days, so oogenesis occurs periodically. It is not continuous. In spermatogenesis, during the growth phase, the primary spermatocytes shows only double the increase. In oogenesis, primary oocytes may show the increase of about 4 to 8 times. In spermatocyte, 4 spermatids are formed from one primary spermatocyte. In oogenesis, only one ovum is formed from one primary oocyte. In spermatogenesis, equal cytoplasmic division during meiosis I and meiosis II, and no formation of polar bodies. In oogenesis, there is unequal cytoplasmic division during meiosis I and meiosis II, and resulting into formation of polar bodies. In spermatogenesis, all stages are completed and the sperms are formed in the testes only. In oogenesis, secondary oocytes leave the ovary and final second meiotic division at fertilization in the fallopian tubule. In spermatogenesis, the male gametes or sperm is comparatively very small, whereas in oogenesis, the female gamete is very large comparatively. In spermatogenesis, spermatids undergo spermatogenesis to become sperms. In oogenesis, no such stage after formation of otid or ovum. In spermatogenesis, it takes longer time to complete. In oogenesis, it takes a shorter time to complete. Question 19. Describe the hormonal control of the post-fertilization changes in the human female up to breastfeeding. Part B. Describe the stages in the process of parturition, that is birth. Part C. How is viability of the sperms in humans maintained? Start with part A. Describe the hormonal control of the post-fertilization changes in the human female up to breastfeeding. Human chorionic gonadotrophin hormone is secreted from the placental trophoblastic cells, causes persistence of the corpus luteum, which secretes progesterone from the luteal body. Continues. Progesterone, together with small but steady secretion of oestrogen from the ovaries, maintain a continual development of the uterine endometrium. Progesterone and oestrogen also inhibit further secretion of follicle-stimulating hormone and luteinizing hormone by negative feedback, as well as further follicular growth and ovulation, respectively. After the first trimester, 3 months of pregnancy, the corpus luteum regresses. The real roles are taken over by the placenta. During the entire course of pregnancy, the level of oestrogen keeps increasing as that of progesterone keeps reducing. Oestrogen promotes, while progesterone inhibits uterine contractions. Progesterone and estrogen allows growth of the mammary glands in preparation for lactation. The two hormones also inhibit the secretion of prolactin from the anterior pituitary gland. A sudden drop in the levels of the two hormones at the end of pregnancy permits the secretion of prolactin, allowing the onset of lactation. In the last moment of pregnancy, oxytocin levels from the posterior pituitary glands rise. Oestrogen increases the uterine wall sensitivity to oxytocin. Oxytocin stimulates the release of prostaglandins that causes cervical ripening, as well as initiating the myometrial contraction. Milk flow from the nipples and its let down is stimulated by oxytocin. Colostrum being the first milk produced during lactation. Part B. Briefly describe the stages in the process of parturition, that is birth. The onset of birth is triggered by the decrease in progesterone and increased estrogen level during the last stages of pregnancy. The posterior pituitary produces oxytocin, which causes the contraction of the uterus that increases in force and frequency. Cervical dilates to allow the passage of the baby's head into the vagina, while the embryonic membrane ruptures. The fetus is expelled in down face position, followed by after birth, umbilical cord and placenta expulsion. Part C. How is viability of the sperms in the humans maintained? Sperms are made at a temperature quite lower than the core body temperature. Sperms are bathed in a thick mucus and milk fluid, the seminal fluid, which provides nourishment and optimum pH. The prostate gland secretes an alkaline fluid that neutralizes the acidity of urine and vaginal fluids, aiding sperm mobility. The seminal fluid contains fructose, an energy-yielding substance for the sperms, aiding spermatic viability. Prostaglandins suppresses immunity of the female against foreign sperms. Proteolytic enzymes contained in the milk alkaline fluid from the prostate gland liquefies the coagulated ejaculate and the cervical mucus immediately after coitus, permitting mobility. Mucus, which form a semi-viscous fluid in which the sperms swim. Question 20. What is meant by menstrual cycle? Part B. What are the effects of uncontrolled birth among humans? And part C. How is growth of the Graafian follicle in the ovary initiated and controlled? What is meant by menstrual cycle? Menstrual cycle is a period of regularly occurring physiological changes in the endometrium of the human female. Some primates, in which absence of fertilization culminates in partial sloughing of the endometrium, manifests as a monthly blood shed, which we call menstruation. has the following phases. There is the follicular phase. This begins from the previous menstruation and ends with ovulation at day 14, during which the ovarian or Graafian follicle develops. There is the proliferative phase. Here, estrogen mediated increase in the endometrial thickening occurs simultaneously with the follicular phase. Then there is the luteal phase. This begins from ovulation to menstruation, usually 14 days long. Part B. What are the effects of uncontrolled birth among humans? 1. Environmental hazards due to overconsumption of natural resources. Congestion associated with higher risks of disease spread. Reproductive diseases, disposition, for example, vaginal fistula, STDs. There is high risk of congenital anomalies, chromosome aberrations, defects such as Down syndrome. There is frequent or unspaced births, adversely effects on the health of the mother and reduces the life expectancy. Population explosion, resulting in resource overexploitation. Part C. How is the growth of the Graafian follicle in the ovary initiated and controlled? Initiated. Primordial follicle, one enveloping the primary oocyte, arrested in the diprotein prophase 1, undergoes primary phase of growth, in which the follicular cells surrounding the primary oocyte changes from squamous to cuboid. They proliferate to produce stratified epithelium of granulosa cells. The primary follicle is formed. When you look here, you will see the germinal epithelium. And we have the ogonium coming off from the germinal epithelium. It becomes surrounded with the primordial follicle, which is going to undergo the primary phase of growth. And those cells, the follicular cells surrounding the primary oocyte inside, will keep on changing from squamous shape to cuboid shape and then to stratified epithelium. And then we say a primary follicle has been formed. Granulosa cells, they also secrete glycoprotein coat on the primary oocyte, forming the zona pellucida. So these cells that are surrounding the primary oocytes, which we are calling the Gronassa cells, they are going to secrete a coating, which is a glycoprotein coating on the primary oocyte and forming a layer called zona pellucida. Many of the primary follicles become atretic. Few viable ones enter the secondary or antral phase. Many fluid-filled cavities emerge in the primary follicle. These coalesce to form one large cavity, the atrum. So when it goes on growing, there are fluid-filled spaces that are seen in the follicle. Those fluid-filled cavities, they later emerge, and when they merge together, they form a large fluid-filled space, which we call the atrum. Connective tissues inside the ovary form the protective sheath around the follicle, divided into highly vascularized thicker internal and less vascularized, more fibrous, thicker external. Then secondary or Graafian follicle formed. Control. Control is along the hypothalamo-pituitary gonadoaxis. Begins with the specialized hypothalamic neuron secreting gonadotrophin-releasing hormone, which stimulates the gonadotrophs of the anterior pituitary glands to secrete the follicle-stimulating hormone, which initiates the growth of the Graafian follicle. Oestrogen from the ovaries inhibits follicle-stimulating hormone release by negative feedback, while stimulating the production of luteinizing hormone, which causes pre-ovulatory changes, in which there is resumption of meiosis I in the primary oocyte. Secondary oocytes and the polar bodies formed. Thank you very much for participating in reproduction in living organisms. Question 16 to 20. Please do like, share, and subscribe.